Okay, so taste and smell are very similar in the fact that they're both going to utilize chemoreceptors. Okay, so the molecules, when we were talking about olfaction, we described them as volatile molecules in which they're interactive. So we call those odorants. Well, here we're going to call those molecules tastants. Okay, so you do want to know that the gustatory cells are chemoreceptors. Those are exterior receptors. They're going to be taking things in from the exterior environment and interacting with them. So your taste buds live in these little bump-like structures on the tongue. And those bump-like structures are called papillae. We're going to go over the different types of papillae. Not all papillae have taste buds, all right, or chemoreceptors. Okay? The first one here, the filiform, they do not. You need to know this. They do not have taste buds. So no role in tasting things. But they do help the tongue move food around in your mouth. Okay, they're like cleats. They kind of cause a grip on food as you're forming that food bolus. Okay, and so you start to move the food around, and you're going to see if this is the tongue here. All right, these taste buds will sit on the anterior 2/3, which is pretty much all of this area here. Okay, because the tongue goes back like that too. All right, so the anterior 2/3 are going to be the filiform papillae. The next, and they're aptly named, are the fungiform, all right, because they look like a mushroom. So they don't have a lot, but they do have a few. And so we're going to see them here on the tip, and we're going to see them on the sides here. Tip and the sides are the fungiform. Okay. Then the next group of papillae are the folate, and then finally the valate. The valate are my favorite, but the folates, okay, think of fall folia, all right, the changing of leaves in the summertime. These are going to have leaf-like ridges, all right. These are going to be effective in early childhood. Okay, again, we're dealing with just a couple of taste buds here. All right, and they're going to be found on the posterior side more towards the back, like back in here. Okay. All right, but the important ones are the ones, and they're easy to remember because they form a V-like configuration. Okay. And they sit along this ridge. I'm not going to try to draw them all in, but you will see when I show you on the other slide. These are the valate ones. That's how I remember them. They're few in number, all right, anywhere between 10 to 12 of them. All right, but this is where the majority of your taste buds are located. It's also located on the posterior dorsal surface of the tongue. Dorsal is the top portion of your tongue. Okay, so when you stick your tongue out at somebody, all right, that's the dorsal surface. Okay. And so these guys here are going to house the most taste buds out of any of the papillae. All right. So now what I want to do is go over the actual anatomy of one of our taste buds here. So if you see here in this picture, all right, of the tongue, you can see here on the tip, all right, you have the filiform, all right, or not on the tip, I'm sorry, on the anterior 2/3, okay, but there's no taste buds in these filiform, all right. The fungiform are going to be on the sides here and the tip. And then we have the folate, the leaf-like ones. You can actually see them on your tongue. If you stick your tongue out far enough, you can see these ridge-like structures. And that's where the folate papillae are. And then you can see right here, here's our nice V configuration. All right, that's the valate. Okay, those guys sit right there. You can actually see those if you stick your tongue out far enough. Okay. All right, so when we're looking at a taste bud, think of the taste bud like Shrek, okay? All right, you know how ogres are like an onion? They have layers. That's what a taste bud has, all right? And if you look at it, look at this thing. It looks like an onion, okay? So again, we have this 3-cell makeup here to this structure. We actually, the first cell listed is the actual chemoreceptor cell, the gustatory cell. All right, this is the cell, all right, that is going to detect the tastings. That's where your chemoreceptors are going to reside. Now notice here, 7 to 9 days. They don't last very long. And so I get people asking me all the time when they get COVID, if I lose my taste of my sense of taste, all right, or smell, all right, those chemoreceptors regenerate relatively frequently, all right, 7 to 9 days for taste. What's going on? How come? Well, the response to that is, well, then COVID is not affecting the chemoreceptors for gustation. Smell, same thing. They regenerate every 40 to 60 days. And the people have my son for a long time. 2, almost 3 months, he didn't have sense of taste or smell. Very good, very well. He couldn't taste or smell very well. And so again, it was one of those things we were just trying to figure out. He just started getting it back. So I was like, sweet. Okay, so the gustatory cells are the cells that house the chemoreceptors, of course, supporting cells. We know what they do. They're going to help support the gustatory cells. Then the basal cells. These are the baby cells. Okay, stem cells that are going to grow up one day and replace the gustatory cells. They just have to wait 7 to 9 days to do so. Okay, so you can see on that Valet papilla here, look at the size of this thing. All right, pretty large. And all the taste buds here are on the lateral sides of the Valet papilla here. So then when we zoom in, all right, you can see you've got your gustatory cells that house the chemoreceptors, and then on each of those gustatory cells, you can see the sensory neurons that are coming off of these cells, and they're all going to come together and form this one taste neuron here. And then at the base, we got our little baby cells. So at the top here, all right, of our taste bud, we're going to have our small microvilli. And they poke through this little hole here, and that's a tasting pore. So when you're mixing up saliva with whatever it is that you're ingesting or chewing on or eating, all right, you break up those food particles, they'll come in contact with these microvilli here at the top portion here. All right, so those microvilli are just dendritic endings. Okay, they extend through that taste pore, they come in contact with the tastings, and voila, then you have your gustatory sense of taste. All right, so we're going to see, you get to go into it in much more detail in chapter 26 when you learn about digestion. You're not really going to talk about taste that much, but you talk about the mechanical and chemical processes of digestion, and it starts there in the mouth. Once the saliva starts to actually kick in, when you have that visceral reflex, do you remember which cranial nerves? This just jumped into my head. Does anyone remember the cranial nerves that will stimulate the submandibular, the parotid gland, and the sublingual, did I say submandibular? Yeah. Sublingual salivary glands? Okay. 7 is going to stimulate the subglands, sublingual and submandibular, all right, salivary glands. Parotid, which is the largest one, that's the one that gets affected by mumps, that's going to be the glossopharyngeal, cranial nerve 9, okay? And both of those, as we know, are part of the parasympathetic nervous system. Remember, 3, 7, 9, and 10? Well, 7 and 9 are part of it, and, all right, parasympathetic nervous system has to do with rest and digest, and that's what we're going to see here, okay? So the tastings are going to dissolve right in the saliva because that's what the saliva is designed to do. It helps to break down certain food products on a chemical level. Also helps to coat the food. We want to make it lubricated so when you swallow it down, you're not tearing apart the inside of your throat, okay, and the esophagus here. So let's talk about the pathway and how we get that sensation of taste from our tongue and mouth to our brain. Because remember, we don't know what the heck what we're tasting until that information, all right, comes to the cerebral cortex, okay? So we're going to start off with our sensory neurons. That's the primary neuron. All right, that's going to extend from those gustatory cells. All right, and it's going to head back towards the medulla oblongata. Okay, now break the tongue down into 2 parts. The anterior part of the tongue, that's going to be the anterior 2/3. It's going to look something like this, like I drew before. All right, this whole area here is going to be innervated by cranial nerve 7, facial. Okay, so this will be 7. And then back here will be cranial nerve number 9. Well, that's easy to remember because both of those cranial nerves, all right, had a role in salivation. Okay? So the posterior 2/3, all right, of the tongue are going to be cranial nerve number 9. My drawing is horrible. I should actually extend the tongue back more. There we go. So 1/3 up front and then 2/3 in the back. Now that's just for taste. Please don't be confused because the general sense of touch for the tongue is different. It's not going to be 7 and 9, okay? So just for taste, all right? So that's what we're talking about here. So the gustatory pathway is going to involve cranial nerves 7 and 9, okay? And so they're going to carry the taste sensations on their primary neuron, all right, all the way to the medulla, where the primary neurons will synapse onto the secondary neurons. Real easy pathway. The secondary neurons go up to the thalamus. Okay? And then from the thalamus, we know this story, it's as old as time. From the thalamus, the tertiary neuron goes to the primary gustatory cortex. Where? In the insula. All right, notice, this one goes to the thalamus. Which one didn't go to the thalamus? Which sense? Remember, we covered it last week. Smell. Olfactory. Olfactory. Smell does not go to the thalamus. You need to know that. I guarantee you. All right, well, I can't guarantee you because it's a randomized test. But there's a good possibility you'll be asked that. Okay? So you should know that. All right? Smell does not go to the thalamus. Okay? It's the only one of our senses that bypasses the thalamus. Okay? So you see the chain here? Primary neuron goes from, sensory neuron goes from the tongue to the medulla. Synapses on the second. Secondary neuron goes from the medulla to the thalamus, where it synapses on the tertiary neuron. Tertiary neuron goes from the thalamus up to the cerebral cortex. Okay? All right, so we have 5 basic taste sensations. When I was growing up, we only learned the first 4 here: sweet, salty, sour, and bitter. Okay, the 5th one here is umami, all right? And umami has to do with a meaty flavor derived from amino acids. Okay, amino acids. All right, so sweet is going to come, you should know this, from organic compounds. Salt comes from metallic ions, and I tell folks, if you ever cut yourself and you lick your wound, all right, it's got a metallic-like taste. Yes, it is from, you do have iron in your blood, but also, all right, you also have sodium and potassium floating around in there too. Okay, it's not just from the iron in there, from the hemoglobin. All right, sour comes from a low pH structure from acids. All right, vinegar is an example. And then bitter is the opposite of the pH scale. All right, alkaloid, something that's basic. All right, has anyone here ever tried unsweetened chocolate before? Oh yeah, it's not what you think. All right, it is not. There was a guy who used to work at Applebee's, and we had a server, not a server, he was a cook. This guy was always making people do, or daring people to do these dares, okay? And one was to eat a whole bar of unsweetened chocolate. And people are like, oh, that's no problem. I can do that. No, you can't. All right? It's difficult. I haven't met anybody that's been able to. I'm sure there's people out there. Yeah. But unsweetened chocolate was nasty.
We had the powder kind of phone.
Okay.
And I would try to, like, as a kid, make chocolate milk and did not understand why it tasted like that.
It's awful.
It's horrible.
Yeah. All right. So for these following tastants, for sweet, bitter, and umami, all right, the tastants are actually going to involve a secondary messenger. Okay. So if you recall, we briefly talked about a secondary messenger system when we talked about olfaction. So basically what we're going to see is that tastant, that molecule, is going to be floating along in the saliva, comes in contact with the gustatory cell. All right, it's going to stimulate, all right, a membrane receptor on that cell. But that tastant never enters into the cell. It stays on the outside. But once we stimulate that membrane receptor, it's going to activate. So here is this membrane receptor, and here comes our tastant here. All right, put a T inside. All right, and this membrane receptor sits on the plasma membrane. So it's going to stimulate and activate the G protein. Here's G. So it's going to stimulate it. I'm going to put like a lightning around it. Now it's stimulated and activated. All right, and that G protein is going to go through and activate a series of enzymes on the inside of the cell. Eventually, it's going to trigger the opening of, well, we're going to see depolarization. So we're going to see the opening of cation channels, which are going to be calcium channels, all right, sodium channels. So sodium, calcium will enter into the cell, causing the inside of the cell to become more positive, less negative, thus causing depolarization. And that's what we're going to see. Now, the nice thing about salty and sweet tastants, all right, because they are ion-based, they can directly depolarize the cell, which is convenient. We don't need a secondary messenger system for that situation. Okay? All right. So once we cause depolarization of the gustatory cells, you know the story. We depolarize the cell, we start to produce action potentials. That sensory cell then sends its action potential messages down the primary neuron. And then that primary neuron synapses on the secondary, which synapses on the tertiary, all the way to the cerebral cortex. That's what we're going to see. So the primary neuron, you need to know this, are going to be cranial nerves 9 and 7 for taste. Okay? Please don't forget that. Okay? Questions so far? Not too bad? Okay. All right, so now I want to get into a little bit more specific, because I'm just being general, and I'm saying, all right, the primary neuron goes from the tongue to the medulla oblongata. All right, so let's talk about that, okay? So the primary neuron goes to the medulla oblongata, but specifically to what we call the nucleus solitarius, okay? So this nucleus, all right, remember, if you remember what a nucleus is. A nucleus is a cluster of cell bodies in the central nervous system, all right? So therefore, you're going to see synapsing, all right? When you see cell bodies, there's most likely going to be some synapses around that area here, okay? So in this structure here, this is awesome, all right? Because that primary neuron is going to head here via the cranial nerves to the nucleus solitarius. That's going to trigger what? Salivation, stomach secretions. All right, well, salivation involves cranial nerves 7 and 9, but the motor component, remember, cranial nerves can be purely sensory, purely motor, or both. Cranial nerves 7 and 9 are both, okay? So they are going to trigger the beginnings of salivation, all right, cranial nerve 9 and 7, and then we're going to start to see, and I'm so excited for you guys because you get to learn this, all right, then you get to start to see the stomach. This is as soon as you're putting food in your mouth. In some situations, if you start to smell food, right, or something, you'll start to salivate, you'll start to increase stomach secretions. All right, that's something different. All right, well, it's not something different. It is something different, but I already talked about that. All right, we talked about olfaction, okay, involving the limbic system. Okay, just right now, as I'm thinking about food, I'm actually salivating in my mouth, and I'm sure my stomach is starting to increase the stomach secretions. What happens here is when you start to put food in your mouth, you're increasing your salivation, you're coating that food. We start to compact into what we call a bolus, right? And it's just a glob of food, okay? As that's going on, your stomach's getting ready. It's prepping itself. It's like, all right, let's do this. We're going to start digesting things both mechanically, which is the actual physical breakdown, and chemically, which is where we're going to start mixing in enzymes and start breaking chemical bonds between these molecules here. So we're going to start seeing that happening. Now, in certain situations, right, if you give my son shrimp, you invoke the gag reflex, okay? So this is important too, and again, it helps with reflexes. We're talking about this in chapter 14. All right, it's a survival mechanism, okay? But it also helps us because, again, if back in the day when we were hunter-gatherers, all right, and you went and you were picking food and it smelled awful, all right, and you gagged, most likely you weren't going to eat that. That's a survival mechanism. Most likely that food was bad, okay? In some cases, it can be psychological, like with my son, because he doesn't like shrimp, and every time he smells shrimp, he starts to gag, all right? And in certain situations, you can have vomiting, okay? From the primary neuron, all right, that secondary neuron gets relayed, all right, from the nucleus solitarius to the thalamus, all right? And then from there, we know the rest. It goes to the primary gustatory cortex, where we have that conscious awareness of what we're eating, what we're tasting, okay? All right, any questions? Oh, let me not forget this too. All right, taste, yes, is affected, all right, if you pinch your nose. All right, if you have a cold. I regretted that, showing that to my children if they're eating food they don't like. If you pinch your nose, you can't taste it as well. Because my wife gets mad when they do that at the table. Okay, and here's the last picture. I love these pictures here. All right, just one more. We'll get into vision here in a second. All right, so here's our fella drinking in coffee, hot cocoa, stimulating the primary neurons here in both the facial and glossopharyngeal primary neurons. All right, that's going to stimulate and send that sensory information here up to the medulla oblongata, all right, into the nucleus solitarius here, okay, which were there. All right, those primary neurons are going to synapse onto the secondary neurons. Those secondary neurons are going to send up to the thalamus. And then from the thalamus, all right, they're going to synapse onto the tertiary, which are going to go to the primary gustatory center. And then that's when you have conscious perception or realization of what the heck it was that you just put in your mouth. You guys think it's going to be weird when everyone's walking around without masks? It's going to be weird. I won't be able to recognize some people because I'm so used to seeing them with masks on. Yeah, isn't that funny? It's so funny you say that because I've had this image in my head of certain people, and then when they take their mask away, I'm like, you don't look like I thought you looked like.
I saw my speech teacher down for the first time the other day.
Yeah. Oh, yeah. Well, normally I have a beard, but I can't wear a beard with this thing on. Yeah, it's awful. All right, let's talk about vision. Okay, we're going to slow things down a little bit. The visual pathways and the visual part of this chapter can be a little bit confusing for folks. Okay, so I'll keep it simple. KISS principle. All right. So when we are discussing this sense, all right, our stimuli is going to, because remember, we have receptors, and the receptor's job is to monitor a stimulus, right? The stimulus is going to be light, and there's three parts to that of the characteristics of light. One, it's going to detect how intense the light is. Two, what color, and three, if there's movement. Okay? So our visual stimuli are going to be what we see. Because essentially, what you're looking at when you're seeing colors or objects, you're just seeing how the light is being reflected off of those structures and entering into your eye. Okay? So don't forget the name of the receptors for vision are photoreceptors. And they have those three characteristics: light intensity, the color, and movement. That's what they're able to monitor. That's what they're able to pick up. All right, so in lab we talked about those photoreceptors, and there's two types, rods and cones, all right? So the rods are going to be, yes, more numerous, okay? But they're going to be the ones that are activated or that we use quite a bit in dim light, okay? So these structures are our night vision receptors. All right, so we're going to see these guys more so on the periphery of the retina. All right, so when you're looking at the retina, remember what I said, you're going to see these more on the periphery because the cones are primarily going to be found at one end of the retina, mainly by the fovea centralis. Remember that structure, that little dot that we labeling? All right, that was on the back part of the eye, okay? So when we talk about the rods here, okay, this is an example of convergence. I don't know if you remember that concept, but you have several structures, all right, that will converge. I'll kind of do a horrible drawing for you so you can see. All right, here's 5 rods, all right, and all their neurons and axons will converge, all right, onto a bipolar cell. Bipolar cell is a cell that looks like this. You have the cell body at one end, and you got one process at one end and another process opposite. One's an axon, one's a dendrite, okay? So that's what we're going to see. We're going to see several rods converge onto bipolar cells, all right? Now, the good thing is you've got lots of receptors that are converging on this bipolar cell, all right, which is great for sensitivity, but not good for specificity, which is how sharp the image is going to be, okay? Great for sensitivity. So when you wake up in the middle of the night from a deep sleep, you've had a nightmare, you're lying there in your dark room, okay? Your night vision is pretty good at that point, okay? So that's what you're primarily utilizing if you have to go downstairs to get a drink of water, or go to the lavatory, go to the bathroom, all right? You're relying on your rods as long as there's really not any lights turned on in the house, okay? So those are going to be used quite a bit at night. Now, cones. This is their home, the fovea centralis. That is where that sharp vision comes in, all right? Now, here's the thing. They're awesome for color, okay? High intensity lights, we'll talk about what happens with that, all right? But they can be activated, all right, by high intensity light, all right? Like we're in here, right? In this room, we're using our cones quite a bit, not so much the rods. All right, the rods are our night vision here, all right? Whereas with the rods, we saw convergence occurring, all right? Several photoreceptor rods will converge onto one bipolar cell. Here we're seeing that one-to-one relationship here. That's going to give us that sharp image, okay? But that's only going to occur when we have enough light for us to see by, like this room. This is considered to be bright light. So we're primarily going to be using our cones here, right? Obviously, we're going to be using our cones too if you can see the color of things. All right, rods can't detect the color. Easy to remember, cones, color, see, okay? All right, so this picture should look, no it's not, I was going to say this should look familiar to you, but it shouldn't because I didn't show you this in the lab, okay? But what we're now seeing, all right, is the retina, okay? And actually, we peeled away all the other cell layers, okay? So this right here, we peeled away these 2 cell layers here, and we're just dealing with the photoreceptor cell layer, okay? So the photoreceptors, all right, our rods and cones, are going to sit right against the choroid here, all right? And they're implanted into the pigment cell. Remember, the pigment cells, they absorb aberrant light, like random light. So it's bouncing around the eyeball. They help to absorb that because they have melanin. And the melanin helps to absorb that. So if we look here at our photoreceptors, all right, so we notice at the one end, I'm not going to say distal or proximal, that'll confuse you. But at this one end here, we call that the outer segment. You can see all these discs here. Okay, in the discs, all right, this is where we're going to have our photopigments. We'll get into this in a second, all right? And it's the photopigments which are going to be the chemicals that are going to help us to detect the light. And I'll bust it all down here, all right? Then we move further into the cell, towards the cell body. We have the inner segment. That's where all the organelles hang out, like the mitochondria, and these organelles that have to do with energy production. Then we have our cell body, and then at the far end, all right, is where we're going to see our synaptic terminals that are filled with neurotransmitter, okay, synaptic vesicles, because it's these structures here, okay, that our photoreceptors are going to synapse on the bipolar cells, all right, and some of the horizontal cells, okay. We're going to see synapses down here, okay. But the interesting thing is that light comes in and it hits these structures first. Doesn't matter. Doesn't do any good because there's nothing in these portions of the cell that light can interact with. It has to make it all the way down here, all right, to the outer segment here where the discs are, where our photopigments are. And we'll talk about what happens with that, okay? So keep in mind, all right, cones, sharpness of vision, color. Rods, dim light, night vision, okay? Questions? Okay. So, remember that term transduction? Talked about it at the beginning of this chapter. Transduction is when you're going to take the stimulus energy and convert it into an action potential. Okay? So we're now going to talk about taking that light energy and converting it into an action potential. And so we call that phototransduction. Okay? So phototransduction, right? You know this, we're going to take light and turn it into electrical signals, which are, in our cells, action potentials. Okay? So the rods and the cones are the only structures, the cells that are able to do this. All right? And it all has to do with what's sitting in the outer segment. All right? The outer segment contains those discs that hold that photopigment. And it's that photopigment that's going to absorb the light energy, and then it's going to cause some sort of configuration, which we'll talk about here in a second, change, all right, in the molecules there of that photopigment, okay? And then we'll elicit some sort of response, all right? So one thing I did not mention about the synaptic terminals here, the neurotransmitter that I want you to be familiar with is glutamate. All right, glutamate plays a big role in neurotransmission, all right, in a lot of neurological structures for special senses and even in the brain. Your brain likes to use glutamate too. All right. Phototransduction. Let's talk about it, because I want to break it down for you here. Okay, so understand it occurs in the outer segment of the rods and the cones, and we're going to deal with this protein, all right, called opsin. And we're also going to deal with retinol. Retinol should be familiar because it comes from carrots, all right, not specific, well, it's carrots, but that's not the only source. It comes from vitamin A, okay? Vitamin A is a fat-soluble vitamin, all right, high, found in beta carotene, okay, but in carrots quite a bit. All right, it's these two, all right, structures that are going to help us with that phototransduction, okay? So let's go down and break it down here, all right? So when we talk about pigment types, all right, we're going to deal with different types of opsins that can process and convert different wavelengths of light, all right, which means different colors. I'm going to come right back to this slide. This slide here shows us the different wavelengths, all right, to light. And it's just the primary colors here, all right? So we're dealing with the primary colors. We've got blue, green, and red. And if you notice, they have different wavelengths. So it's those different wavelengths that are going to trigger, and even rods too, all right, they can see at a specific wavelength. So it's these different wavelengths, all right, we'll get into this, which are going to actually cause the change and affect these photopigments. So you have a specific photopigment for red wavelength. You have a specific photopigment for the blue wavelength, all right? So blue wavelengths can't affect red cones. See what I'm saying? It's specific. That's how your brain knows. Whatever cone is activated, that means that that's that color, okay, when we're talking about the color, okay? All right, so keep that in mind. Each photoreceptor has only one photopigment type. So blue cones are good just for the color blue. Green and red, nothing. Nothing. They will not be activated. Okay? So rods, their photopigment is called rhodopsin. And we're gonna go on that for a little while. We'll come back and talk about cones here in a moment. All right? So the photopigment is rhodopsin. Okay? In our cones, all right, our photopigment is called photopsin. Okay? So you have 3 different types of photopsins in cones, and that is dependent on, all right, what the cone is for. Is it red? All right, is it blue? Is it green? All right, and they're all different. All right, so blue has a relatively short wavelength. All right, so the blue cones will only be affected by a 420 nanometer wavelength. All right, for the blue spectrum. Okay, so it's just different. If you want to keep it simple, blue is short wavelengths, green are going to be medium or intermediate, and then reds are going to be the long wavelengths. Okay, but just keep that in mind. Questions about that so far? All right, I'm trying to keep it simple. All right, when we're talking about our photopigments, all right, we have 2 structures involved: the opsin protein and then the light-absorbing retinol that comes from vitamin A. Okay, so our photopigments in rods is called rhodopsin, and then our photopigments in the cones are photopsin. Good so far? Because we're going to start to get, I won't say goofy here, but we're going to start to get a little bit more in detail. Okay, so before I go on, and we're talking about colors anyways, I want to introduce you to my father-in-law. Here's a man that is colorblind. It's X-linked recessive. Okay, what does that mean? If you've taken a genetics class, all right, folks that are born female are XX chromosomes, folks that are born male are XY. So if you have a recessive condition, you have to knock out both X chromosomes. Well, folks that are born men only have one. They're XY. So you just have to knock out one. All right? Folks that are born female, you got to knock out both of them. That's why you're going to see it more often in males. Because it's easier to knock out one than 2. Okay? So doesn't mean if you're colorblind that you're going to be completely devoid of being able to see colors. You can. If it's absent, yes. All right? My father-in-law is deficient. So he can see certain shades, he can see browns and some reds. That's one of the reasons why. Remember the traffic lights? If you go up to a traffic light, there's 3 lights. All right? Which one's the green light and which one's the red light? You sure? Green is the bottom. It's funny. Ask that to people. A lot of people don't know. I shouldn't say, you don't think about it. We all drive every day. Yeah, you never, you know. Well, in some older towns, they used to put the traffic lights on the side. So no one had a freaking clue, especially if you're colorblind, all right, if it was red or if it was green. So you don't see them, except in older towns, them on the side anymore. That's at least in New York. I can't speak for everyone down here. Okay? So when you are affected with color blindness, it's going to usually be these 2 colors, red and green. Okay? All right, let's get back to business and talk about that phototransduction. Okay? So we're going to start off talking with the rods. All right, I'm going to go nice and easy. Okay, so you're in a dark room at night. Okay, like I said, I'm going to keep using that example. You wake up in the middle of the night, pretty good night vision. All right, so right now, your photopigment, all right, rhodopsin, has this configuration. We call it the cis-retinal configuration. Okay, so in a dark room, you have the cis-retinal. So I'm going to come back to this slide. Okay, so here, all right, here's your photopigment. Okay, you got the retinal molecule on the opsin protein here. Okay, so combined, that's rhodopsin. So in a dark room, it's in the cis-retinal configuration. So look, all right, the retinal molecule is just bent. That's all that it is. Okay, it's bent. Okay, you can see it right here. And in fact, it's bent at 2, 3, 4, 5. It bends at between the 5th and 6th carbon. Okay, so when light strikes it, okay, so when light strikes the photopigment, it is going to convert, all right, the cis-retinal, molecule into what we call the transretinal. Look what it did. It just straightened out that bent molecule. That's all it did. All right. So the actual light rays, all right, when the photopigment absorbs that light, just changes the cisretinal to the transretinal. That's all we're seeing there. Okay. So because of that, all right, the retinal molecule pops off the opsin protein. It's like, I'm out, peace out, all right, I'm gone, all right? And this causes, all right, what we call the bleaching reaction, in which, if you've ever been in a dark room, perfect example, you've been in a movie theater, okay, movie gets over, and instead of walking out through the main lobby, you go out through the back door, right into the parking lot, and it's still afternoon, and that light is bright, all right? So you walk out and you're like, oh my gosh, and you see a lot of white spots. That's the bleaching reaction, all right? Because you've caused your cisretinal to change its configuration, and it pops right off of the opsin protein, all right? So remember, what is the job, all right, of our rods? Night vision. Well, you just stepped out into the sun. You're not going to be using your night vision, all right? You've caused this bleaching reaction to occur. So what we have to do now is we've got to rebuild our photopigment. So we've got to take this transretinal and we've got to convert it back into the cisretinal. No problem. That's not a big deal. It just takes a little bit of time. All right? So that cisretinal, all right, is what we need. The transretinal gets transported into the pigmented layer. What's the pigmented layer? Right here. That's right here. So all that has to do is it has to leave this disc here, all right? The discs are here. It's just going to go into the pigmented layer for a moment, all right? And we're going to convert it back into, all right, that we're going to convert it from the transretinal back into the cisretinal, okay? So what we'll do is, that'll occur. We transport it back into the rod, all right? We combined it back with opsin. And now we've got a functional photopigment molecule, okay? But unfortunately, it's not going to work in bright light, okay? So keep that in mind. That's what happens when you're, like the song, blinded by the light, all right? So you're going to have this bleaching effect, except in rods, because they're more sensitive to light, because they're best equipped to work in dim light. All right, this process, this bleaching reaction, takes a while. Okay, it can take up to 20 minutes. All right, same thing happens in cones with their photopsins, but it doesn't last as long. Cones are much better suited, all right, for what we consider bright light. Okay, so if you really want to blind the cones, you've got to be, it's like almost, I'm not going to say standing in front of a car and having them turn their high beams on you, but you know what I mean. It's happened. All right, you have to be in a really bright area. Okay, so again, we're going to see the photopsin, which is that photopigment molecule. It will regenerate quicker, all right, in the cones than it will in the rods. So let's take a quick peek here. Check it out. All right, so let me finish my story. Okay, so the sun hits the rods, causes that configuration change of the transretinal. All right, so because of that, it pops off. Okay. So the opsin is naked and afraid right now because it no longer has the transretinal with it, because it's getting transported into the pigmented layer. While it's there, all right, we're going to bend the transretinal molecule back into the cisretinal configuration. Of course, that takes a little bit of ATP, no big deal. All right, once it returns back to the cisretinal, it will then come out of the pigmented layer, back to the opsin molecule. We slap it back on the opsin molecule, and now we have a functional photopigment molecule. So when you are in dim light, it's going full bore. Okay, just don't turn the light on. That's why you ever notice, all right, most of you are pretty young, so you don't have to worry about this, but you get up in the middle of the night, you know, at least for me, I'm kind of hesitant to flip the light switch on if I have to go to the bathroom, because I don't want to be blinded by the light, you know? And so because it takes down my my rods a tad. So I'll just pee in the dark. And that's why my wife gets mad at me when I'm peeing on the toilet seat. Kidding. I never do that. I always sit down when I pee. Just joking. All right, here we go. So let's talk about dark adaptation, all right, and light adaptation.
Which part were you talking about?
All of it.
Okay.
I never wake up in the middle of the night. No, I do. I do too. See, this is usually my last bit of liquid for the night.
It sucks 'cause it's usually like at 5 o'clock and I get up at like 7.
So you're close.
So it's hard to get back to, like, that last 2 hours is just not good.
Right, right. No, I hear you. I hear you. Okay, so when we talk about dark and light adaptation, this is, again, what we're going to see, all right, following the bleaching reactions, okay? So for dark adaptation, when we're talking about the rods, it takes 20 to 30 minutes, again, in a dark, dimly lit environment here, okay? So we just bleached the rods, we just shot some bright light at them, okay? You turn the light switch on in your bathroom when you go to the bathroom, all right? You go into your business, you get back into bed, you lay there in bed with your eyes open for a little bit, and you're going over your school notes in your head or whatever. But if your eyes are open, you notice that you can't see too well in the dark room, all right? But if you're awake for 20 to 30 minutes, you'll notice that your night vision has improved, all right? And that's how long it takes to regenerate that rhodopsin. Now, with the light adaptation here, all right, now you're going to see some other issues or processes occurring here, all right? So when we go from a low light scenario into bright light conditions, what's the first thing you see? Pupil constriction, okay? What constricts the pupil? Parasympathetic nervous system, all right? The constrictor muscle there causes the pupil to decrease in size, all right? Letting less light in, all right? But what will happen is pretty much the cones are going to get hit first, all right, because they're right there by the foveal centralis. The rods are going to be more on the periphery there, okay? So when we're talking about light adaptation, that's a much shorter period of time, 5 to 10 minutes, okay? All right, let's talk about now, remember, we're still on phototransduction. I haven't even gotten to the part about how we generate these nerve signals. Now we're going to talk about that, right? Talk about the bleaching reaction, right? What happens when we expose certain photoreceptors to certain levels of light. But now let's get into what you all have been waiting for, all right? How do we make nerve signals? So again, we'll start with the rods. I'm going to go back and forth between some slides and show you some things. Part of me wants to say to you folks, just abandon a lot of what you've learned so far in this course to understand this, but I don't want to say that, and I'll show you why. Okay, so resting membrane potential, negative 40 millivolts. Keep that in mind, all right? It's a different value than what we've seen in muscle, different value than what we've seen in nerves here, okay? So that's the value that we need to achieve for depolarization, okay? So guess what? In the dark, look at the statement, rods are depolarized. They're depolarized. So when you're lying there in bed, all right, and you wake up in the middle of the night, you go to the bathroom and you haven't gotten out of bed yet, your rods are depolarized, all right? So what does that mean? What's going on, all right? This molecule, cyclic, all right, guanosine monophosphate, or as we like to refer to it as cyclic GMP, is going to be constantly produced, okay? What does it do? It keeps the cation channels open. All right, we know, I just went over that before with you. Cation channels, calcium channels, potassium channels. All right, this cyclic GMP keeps those channels open and allows for those cations to move into the cell to keep it depolarized. All right, so when sodium and potassium, sodium and calcium enter into the cell and they depolarize the cell, we refer to that as the dark current. All right, kind of like the dark web. All right, but we call it the dark current. Okay, and this causes the voltage-gated calcium channels, all right, down in the synaptic terminals of these rods to stay open. Well, if they're going to be open, then you can see our neurotransmitter will be released. We know what happens when you open voltage-gated sodium channels down by the synaptic end of our neuron. All right? It causes neurotransmitter to be released. So this is happening all of the time. All right? So this glutamate neurotransmitter is constantly being released by the rod onto the bipolar cells. Well, guess what? It causes hyperpolarization. All right? Hyperpolarization. Okay? And because of this hyperpolarization, all right, we know what depolarization is. All right, we're making something less negative or more positive. Well, with hyperpolarization, we're making something even more negative. Okay? So if we're making it more negative, we're moving it away from the threshold value. So guess what happens? The ganglion cells are prevented from being excited. Well, then the story ends there. It ends there. Because it's the ganglion cells, all right, whose axons, all right, form the optic nerve. So we're stopping an action potential before it actually can leave the eyeball. Okay? All because, thanks glutamate, all right, it hyperpolarized the bipolar cells. Okay? And because it hyperpolarized the bipolar cells, we can't stimulate the ganglion cells. Because the ganglion cells are the cells, all right, that are going to transmit that action potential to the optic nerve. Let me show you in a picture here. The dark current. It always makes me laugh. Okay, so here you can see, all right, light switches off. You're lying in bed. Okay? So, our cyclic GMP is constantly stimulating the cation channels to open and allow the flow of calcium and sodium cations into the cell, okay, causing it to depolarize. So as long as this cell is depolarized, it is going to cause the release of the glutamate neurotransmitter onto the bipolar cell. And as that glutamate neurotransmitter is stimulating the bipolar cell, it's going to hyperpolarize that cell. And it prevents the bipolar cell from stimulating the ganglion cell. Okay? This is what happens in the dark. Right? So now, let's flip the switch on. Okay? And now we'll talk about what happens when we flip the switch. Do I need to go back to here? Okay, let's do that. Here we go. Did everyone get all this information here? We good? All right, so now we know what happens in the dark. So let's find out what happens in the light. Okay, so a little bit of different situation. So I'm going to walk you through it nice and slow. Okay, so when the light is flipped on, okay, the light will actually cause hyperpolarization of the rods. All right, so here's what happens. All right, we already saw, all right, with the bleaching reaction, what occurs. All right, so when the bleaching reaction occurs, it causes the splitting of the rhodopsin there. All right, but also what will happen is now we've activated, secondary messenger system. This is the third time, all right, that we've talked about the secondary messenger system. We talked about it with olfaction. We just got done talking about it with gustation. And now we're going to talk about it here with our visual system, okay? We're going to activate that secondary messenger system. Remember what I said? Secondary messenger systems involve something on the outside of the cell, stimulating something in the plasma membrane, and then activating something on the inside of the cell. That's what happens. All right, we're going to activate this ASE. What is that? That's an enzyme. That's right. Booyah. Phosphodiesterase. All right, this enzyme, all right, is going to activate and break down cyclic GMP. All right, that's the problem. Remember, what was cyclic GMP doing in the dark? It was opening up those cation channels. That's cool. That was good. We wanted that, all right, in the dark, all right, because it depolarized the cell. Well, guess what happens here? It breaks down the cyclic GMP. The cyclic GMP can no longer keep the cation channels open, so now they all close. And when those cations close, or channels close, no more sodium, no more calcium coming into the cell, so we can't depolarize the cell. Guess what happens? The cell is now more negative, okay? And if the cell is more negative, it's further away from threshold value, we're not going to be able to open up the voltage-gated calcium channels down by the synaptic terminals. We can't release glutamate. Bye-bye, glutamate. Because remember, glutamate was getting released onto the bipolar cells and was causing those bipolar cells to be hyperpolarized. So they couldn't stimulate, all right, the ganglionic cells. Well, guess what? That's not happening now anymore, okay? So our bipolar cells are no longer inhibited. So now they can release their neurotransmitter, well, which is glutamate, and that glutamate from the bipolar cells can now excite the ganglion cells to send their action potential along their axons back towards the optic nerve, out of the eye, back towards the brain. Okay? This is a little bit more involved than one would think. Okay? You guys are doing great, though. You mind if I take a sip of water?
Your bladder.
No joke. I just got done teaching that reflex last night, the storage reflex and the mixturation reflex. I've been drinking a lot of water lately. Okay, so let's see what happens in the light. Blinded by the lights. Wrapped up. Okay, here we go. So now we turn our light switch on. Flip the switch on. Okay, we're dealing with our rod here. Okay, so that bleaching reaction occurs. Okay, we see that cis, all right, retinal configuration changes. It pops off our opsin. All right, all that fun stuff happens, and that is going to trigger that phosphodiesterase enzyme, okay, to start breaking down the cyclic GMP. If there's no cyclic GMP available, because that was the thing that in the secondary messenger system that was opening up those calcium channels, those sodium channels to let calcium and sodium in. Well, those are now closed, okay? So now, all right, the cell is no longer depolarized. It stops releasing its neurotransmitter because those voltage-gated calcium channels close down. So it's no longer releasing its neurotransmitter, so it's no longer hyperpolarizing the bipolar cell. The bipolar cell can now release its neurotransmitter, okay? Its voltage-gated calcium channels open up, okay? I mean, not voltage-gated, they're just regular calcium channels. They open up, release glutamate, its own neurotransmitter, and now stimulates the ganglion cell, all right, to now transmit action potentials through the optic nerve back to the brain. And that's how we're able to see the light, okay? All right. I'm sure there's lots of questions on that, but you just don't know them yet until you go over the material. I get it. How about this picture? You want to be weirded out by something? How come that spoon looks like that? That's funky. That's refraction. All right. What's refraction? Refraction is the bending of light rays. And that's what we're going to talk about now. Okay? So when we talk about how light rays enter into our eyes, right, they get bent. And there's a couple different places that they do. But part of that property of bending of light, that's this refraction here. You can see how the spoon, when it's in the air, looks different. But then when it's in the water, all right, it looks different also. I shouldn't say different. How the spoon, the handle of the spoon looks one way, but then in the water it looks a different way. That's what I should have said, right? And that's based on refraction, okay? So what we're going to see is that light, when it travels through the air, when it travels through a solid, when it travels through a liquid, it gets bent. And when it gets bent, it's going to change the image of whatever we're looking at. And so that's what we're going to talk about. So when we're dealing with our eyes, light has to travel through the air to get into our eye. Then it has to travel through our cornea, which is a solid, so it will bend it a little bit. Passes through the aqueous humor, remember that fluid that's produced in the anterior chamber. Okay? Passes that, nothing really, that's not really too significant. The lens is where we get a lot of refraction. So we're going to bend that light when it goes through the lens. Then it's going to go through the vitreous humor, that goo that sits back there in the posterior chamber. There's going to be some bending there. All this is for a reason, because we need to focus those light rays. Our goal is to focus those light rays on the fovea centralis. That's what we want. That's where the sharpest vision is located. That's where the majority of our cones are located. Okay? So we're going to see, all right, how, all right, when light passes through these different structures with different densities, all right, how it bends, all right, and what occurs. All right, so a couple terms here, all right. We have our near response, convergence of the eyes, accommodation of the lens, and constriction of the pupil. All right, so these are all things that we're going to talk about when we're looking at object or as light comes off of objects at different distances. Okay, so when we talk about the near response, all right, this is when you're looking at something that's 20 feet or less away from you. Okay, so here's what's going to occur. The eyes are going to converge. And that's easy because when you're driving in your car and you're looking down the road, your eyes are pretty much straight ahead. All right, but when you have to look down, like at your watch, let's say, all right, you're going to converge the eyes. They're going to move more medially. All right, and what muscle makes your eyes look medial? Do you remember? Remember those muscles of the eye, the extrinsic eye muscles? What's the one that sits on the inside of the eye? Medial rectus. The medial rectus is going to pull, all right, the eyes. It's not the only one, but it's going to play the largest role there, all right? It's going to pull the eyes inward, all right? And so what we'll see is, all right, as both those eyes converge, we are going to direct the image of whatever we're looking at onto the fovea centralis of each eye. That's our goal, all right? So unfortunately, if you have certain conditions, for example, myasthenia gravis is a condition in which you get weakening of the muscles, you can get double vision. Right? So anything that has weakness of the eye muscles can cause double vision, which is what we call diplopia. Okay? Accommodation of the lens. We've talked about this in lab. All right? This has to do with the ciliary body and the ciliary muscles, the lens and the suspensory ligaments. Okay? So the ciliary muscle here will contract. What does that mean to the suspensory ligaments? They loosen, all right, or slacken. And that causes the lens to become thick. It goes like this. It goes from a thin, all right, to a nice big, thick configuration. Okay? And that's going to increase the amount of bending of the light as it enters in through the lens. Okay? And then finally, the last thing is constriction of the pupil. Okay? We're going to constrict the pupil to let less light in so we can focus more of that light on the fovea centralis. That's where our sharpness of vision is going to be located. The more light that you let in, you can have some blurriness there, okay? Because if it hits the periphery of the lens, all right, it won't necessarily nail the fovea centralis, and the peripheral photoreceptors aren't going to have as sharp a vision because remember, what's on the periphery of our retina? Rods. Rods don't have sharp vision, all right? Cones do. Cones are going to be more located in the fovea centralis, okay? So let's talk about, all right, so that's our near response, okay? For looking at objects that are further away, right, we don't have to have convergence of the eyes, right? And we go from that thick lens to a nice thin lens because the ciliary muscle relaxes, but the suspensory ligaments tighten. And when they tighten, they pull on the lens and make it really, really tall, okay? But at the same time, the pupil is going to dilate. It'll go from this to like this. Okay, that will let in more light, but unfortunately, we'll get a little bit of a blurriness going on. All right, let's check it. Ready, and on your mark, set, go. All right, so the shape of the lens, all right, is going to be different depending on what you look at for certain distances. So, looking at the mountains here far away, all right, we're going to see the pupil dilate to let more light in. All right, the lens is going to flatten, all right, because the ciliary muscle is going to relax, but that's going to create tension on the suspensory ligaments. All right, so we're not going to have as much refraction, okay, and bending of the light rays as they enter into the eye. Now, when you look from the mountains down at your watch, then you're going to undergo accommodation, all right? That is going to cause constriction of the pupil. You'll also see, all right, the ciliary muscles contract, which causes those suspensory ligaments to slacken up and allows the lens to kind of rebound into its thickened configuration, becomes more convex in fact, and that causes more bending of light rays, but more towards the fovea centralis. Our goal is always to hit the fovea centralis there. All right, cataracts. Don't like these things, all right? They are when you start to get these small cloudy opacities within the lens, okay? Several causes can occur, one of them being drug use. Believe it or not, usually you think cataracts are going to be something in an older person. All right, I've seen it in infants, and usually if it occurs in infants, it's because the mother was doing a significant amount of drugs during pregnancy. All right, normally it is an aging thing, okay, but like I said. All right, so the problem is accommodation, looking at things close up, all right, and because you have these opacities there, it's going to interfere with light entering the lens, all right. It can't obviously travel through the opacities, so you're going to have a reduction in visual clarity, and then also you're going to get the intensity of certain colors, all right, or certain images will be reduced there, okay. So a lot of times they'll just remove the opacities. One of the techniques that they use is phacoemulsification, right? Basically, a lot of it is lasers now, but you can use sound waves, just blast those opacities out. There's actually, believe it or not, there's quite a few different techniques that they've been using. All right. Oh, we're close to being done, but not yet. Let me just run through and talk about the visual impairments, and then we'll be done for the day. All right. So this is what I wish I had. I wish I had metriopia. I don't. And as I can see, some of you guys don't either. All right. That's the glasses. All right. But believe it or not, we're going to go through 4 different visual impairments. And I have 3 out of the 4 up here. You know, so I'm a genetic freak. All right. Anyways, hyperopia. All right. If you are far-sighted, all right. And I always thought this is crazy how they describe this. A far-sighted person is somebody that has trouble seeing stuff that's too close. All right. But basically, it's because the eyeball is too short, and what will happen is, all right, that when the rays, the light rays come together and to that focal point, all right, they're going to basically miss their mark, and they're going to go past, all right, the point. Let me show you. All right, here's metriopia. Okay, so you're looking at whatever, all right, and the light rays of whatever it is you're looking at converge perfectly here on the fovea centralis, or what we call the focal plane, that exact spot where it should be. All right, now, all right, if you have hyperopia here and it's not corrected, all right, whatever you're looking at, they actually converge past, all right, the fovea centralis. Okay, so they haven't converged at the correct point. All right, so what we do is we give you some corrective lenses. In this case, a convex lens that starts to bend the light rays before that ever gets to the eye, hence glasses, okay? And then it bends the light rays just before it goes through the lens and the other structures there to the focal plane on the correct area there, okay? All right, so that's hyperopia. All right, so again, convex lenses. Here's me. I'm myopic. All right, myopia, nearsighted. I have problems seeing things far away. So apparently my eyeball is too long. All right, so what will happen is the light rays will converge in front of my focal plane. All right, it'll be too short. So the corrective lenses that I have to wear are the concave ones. So that's this guy right here that you're seeing. All right. Here it is, uncorrected, so the light rays converge before the fovea centralis and my focal point, or on my retina. All right, so it'll again be blurry. So what we do is get me some concave corrective lenses, and they'll bend the light, but just spread them out slightly. By the time they get to the lens, they get bent appropriately onto my fovea centralis there. All right, the last two, because I'm old, I have presbyopia, all right, but astigmatism, all right, is partially, all right, when one eye is shaped abnormally. I was told by one of my eye doctors that my eye is in the shape of a football. That was how he described it, all right? Point being is when the eyeball is shaped abnormally, you have these unequal curvatures there, which are going to mess up the refractive surfaces, okay? Although my astigmatism is improving, all right? And when I first started reading about it, I kept saying astigmatism, but that's not how you pronounce it. It's just astigmatism. So you have astigmatism. All right, and then as you get older, this is the one where you start to see folks wearing glasses, all right, as they get older, presbyopia, okay? And this is basically because the lens loses its ability to shape change as easily. Remember, as we get older, all right, we start to produce fewer and fewer collagen fibers for our skin, fewer and fewer elastic fibers in our skin. Well, our lens is no different. Our lens has a lot of elastic fibers in it. It doesn't have that anymore, okay? So the lens has, it's not as easy for it, all right, to become spherical, all right? So it makes it difficult for the bending of those light rays when we're looking at something up close for us, all right? Surgical techniques, LASIK is one of those techniques that they have, not so much for presbyopia, but more so for folks. I've got a friend of mine, he got LASIK in his 20s and he's still got close to perfect vision. He's almost 50. Anyways, so that's it for today for the lecture material. Let's take a break. All right, and then when we come back on Thursday, we will finish up the visual pathways, and then we'll do auditory and the equilibrium. Okay? All right, I'll see everybody in the lab section.